Thank you, Vincent. I hope you all hear me well. So we will present to you, and I will present together with Shani, a new method, as Vincent said, for whole plant physiological identification and quantification of disease progression, which is performed on the Plantarray system from Plant-DiTech. We just have some technical difficulty. 1 second, please. Okay. So I'm Keren Moselion. I am the CEO and partner of Plant-DiTech. It's a company from Israel. And Shani, who will present together with me, is a PhD student from the Faculty of Agriculture at the Hebrew University of Jerusalem from Israel. And we will both talk about this subject. I will first introduce Plant-DiTech and the Plantarray technology. And after that, Shani will provide more of the research and application that she is doing during her research about disease progression. So let's start by introducing Plant-DiTech. So Plant-DiTech introduced the Plantarray platform, the Plantarray system. It's a high-throughput physiology phenotyping platform. It initially was designed for abiotic stress studies and gained already about 70 scientific articles that were published based on the technology. Some of the customers that are in our customer base are presented here, and we have many more, of course, around the globe. I will describe a bit to you the Plantarray technology for those of you who don't know us so far right now. So the Plantarray technology could be envisioned a bit like a smartwatch, only for plants. So what we do, we measure the plant dynamics, so the continuous performance of the plant physiology in response to different environments. And we do that together for many plants in a randomized structure, as you can see in the picture, for the different treatments. And we measure the soil-plant atmosphere continuum. You can see some of the graphs on the right as an example for that. The technology is based on lysimeters or mini scale, very precise mini scales, but we took it several steps ahead, and it's a much more advanced technology than the maybe lysimeters that you know from the old tech. So this is a more detailed description of the system. What you can see is that on the left-hand side in the image is that we are providing personalized treatments, like personalized cocktails for the soil water for each and every plant, could be different. And also we can apply multi-stress, multi-treatments to each and every plant separately and all together, depends on the design of your experiment. In the center of the image, you can see the soil plant atmosphere continuum measurement. All the traits in green describing the physiological measurement, and all the measurements in blue are the environmental sensing, the soil, the atmosphere. We measure the plants about 500 times a day, so it's on a continuous basis. And all of these measured traits and parameters the measurements are being uploaded to a cloud software where it's all being in real time, being analyzed statistically already. So you can see some of the graphs on the right. The idea about the physiological parameters and why we are working with physiological parameters for two reasons mainly. One is that the stomata is very fast to react to the stomata activity, is immediately reacting to any changes in the atmosphere or any stress at all that affect the stomata, of course. And also all these physiological traits that relate to the stomata or also the biomass gain and other traits that we're measuring are directly correlating to yield. So this is helping us a lot, and this is not, we didn't invent this, this is known in the scientific literature, but we are utilizing this in order to shorten the process for screening. So it's fast screening based on the yield potential between the plants. The measurement that we are taking in a comparative manner simultaneously to all the plants. Very quickly, we can identify those that have more drought tolerance or any other traits or any other ability that we are looking for as a researcher. And we are shortening the whole cycle dramatically. We don't need to wait for until yield. And of course, also the depth of the knowledge is increasing dramatically. So it's not the guesswork anymore. It's much more precise and much more informative and quicker. This is just an example of, let's say, a normal graph from our system, from the Spark Analytics software, where you can see the soil water content that we are controlling. So this is controlled by the system. It's actually, you can see that the drought reduction is in the same rhythm, same rate. And this is thanks to the feedback irrigation that we have in our system, a quite unique capability. In terms of how we control the soil water content, we can control it by the soil sensor, we can control it by on-plant transpiration and other ways. Also, you can see then, the different lines, you can see how the different lines behave, the different varieties of tomatoes in this case, very clearly with statistical analysis already. This is another resolution looking on a day in variations. Here you can see the continuous measurement over a day. In this case, it's transpiration and stomatal conductance versus the environment, VPD and PAR. And every window of difference, let's say, if you can see a peak here, could end in accumulated effect of about 15-20% for just a peak that looks like for 1 hour or 2 hours. So this accumulates very significant yield impact. The system can work with large variety of plants, from Arabidopsis through vegetable crops, field crops, and also even trees, as you can see here. And there are different applications that are useful to do on the system. It's genetic screening, photovoltaic stress, you can change drought. Also, we can control flood, salinity, and so forth. Physiological studies of different kinds, nutrients and biomass screening, seed treatments, and how they affect the whole plant, optimization of agronomical treatments, and biotic stresses. And with this, because we're going to focus the rest of the talk about biotic stress, I'm going to transfer the discussion to Shani. So I will stop sharing and Shani will share from her computer. Forgive me for a second. Shani, it's...
Okay. Hi. I will present to you a section of my research using the Plantarray to identify and quantify disease progression. My name is Shani. I'm a PhD student at the Hebrew University in the Department of Plant Science. I've always had a deep love for plants, but I have to admit I've unintentionally become quite good at killing them in all sorts of ways. But funny enough, the lack of causing stress to plants actually is the focus of my PhD research. Let me introduce you to one of the top 10 fungal pathogens, Moselion mucosisporum. This form of specialis is a soil-borne fungus that specializes in attacking tomato variants. It causes the disease we call Fusarium wilt. The infection starts at the roots and moves up into the xylem, where it blocks the water flow. This leads to wilting, yellowing of leaves, and death. Moselion mucosisporum can devastate tomato yields. It can impact both commercial farms and home gardens. all of our Fusarium work was done in collaboration with Dr. Shai from the Department of Phytopathology. To inoculate our plants with Fusarium, we use a common protocol. 3 weeks old seedlings were cut at the roots and submerged in the carnegie suspension of million kinesiads 2 ml. Then they were planted in 4 liter pots on the Plantarray system. How do we assess the Fusarium wilt disease? For the first few days, you can't see anything in the inoculated plants, like this picture. This picture was taken 11 days after the inoculation, where you can see this leaf is missing, this leaf is yellowing, and some leaves are not fully open. As a scientist, I need to quantify this tiredness and yellowing. How do we do that? Looking at the literature, there are 2 main ways to assess diseases. Usually, an experimenter visually inspects the plants and gives them a score. 0 means no symptoms, and 1, 2, 3 indicate various degrees of yellowing. However, this method can be inaccurate since it's subjective, and I've seen some works talking about the problematic of visual subjective scoring. The other main method is measuring yield or biomass at the end of the experiment. However, I feel this misses some of the disease dynamics because it's only at the end, like 1 point. We want to offer a third option, using the Plantarray system to assess the disease. By definition, disease is an impairment of the normal state of a plant that interrupts or modifies its vital functions. We have a tool that can continuously monitor vital function of a plant. This includes biomass of the plant, transpiration, the water loss, nutrient use efficiency, stomatal conductance, root activity. It's easy to use. It's quantitative and objective. It's comparative. We can have control and infected plants simultaneously on the system, and it's continuous. From day 1, we can follow the disease. Our goal is to use the continuous measurements to detect the disease in plants early, even before visual symptoms. Additionally, we aim to use the system to quantitatively assess the severity of the disease. Before I dive into the experiments we did and which Fusarium strain we used, our tomato cultivar, I want to present to you this overview. This is a summary of the experiments we did. Every row represents an independent experiment with a specific strain of Fusarium and tomato cultivar. In all the experiments, we followed with the Plantarray the transpiration and the plant weight, and we came to inspect the plants visually to see if we see any morphological symptoms. In the table is when t-test showed that the inoculated group is different from the control, same cultivar but with water. In all the experiments, you can see that transpiration was the first to be different between the groups. Next was the weight. And last, we saw visual symptoms, if we saw at all visual symptoms. Fusarium grows in the vascular system and blocks it. So it makes perfect sense that the first symptoms we see are in the transpiration. Just from this overview, you can understand that the Plantarray system gives us an ability to early detect the disease. To cope with Fusarium in the field, farmers and growers use non-susceptible cultivar. Let's talk about plant susceptibilities. There are four main words to discuss it. You can say immune, resistant, tolerant, and susceptible. The problem of some of these words are not black and white. How tolerant is a plant? How susceptible is it? We suggest talking in precise percentage of yield, and I'll give an example. This is an industrial cultivar named M82 that is tolerant to Fusarium. And this is Rehoboth 13, which is susceptible to the pathogen. We followed the plant weight after the inoculation, and we could see differences between the cultivars. You can see the trend starting very early on, but we would look at this stage. At gray, M82 control-treated plants get to around 220 grams. When inoculated, it loses about 15%. Interestingly, Rehoboth 13 in black, the susceptible cultivar, produces higher biomass, 23% more than M82. In brackets, I would say this is not significantly different. But when inoculated, R13 plants decrease biomass in 60%. There is a high correlation between biomass and yield. And now that we have this precise percentiles information, a farmer could decide which cultivar he wants to use. If he wants to take a risk and grow R13 and perhaps gain 23%, or he knows he has fusarium in his field and he is satisfied in losing only 15% with M82. This tool is quantitative, exact numbers, and it's early on. You can see the trend starting early on. We did this with 2 other cultivars, and Marmond Verte, I hope I'm saying it okay, a susceptible cultivar, and Mutella, a resistant cultivar. We followed the transpiration of these plants, and here too, the resistant cultivar in gray is acting nicely. When infected, it loses some transpiration. The susceptible cultivar in black doing better when uninfected, but losing much more when inoculated. I can present this graph one other way, taking the daily maximum for each day and for each plant calculating the relative transpiration. This graph, relative transpiration from the daily maximum. I found this presentation very interesting because you can see nicely how the percentiles stabilize and perhaps how the system can help early on quantify the differences between the cultivars. I talked about differences in tomato cultivars. Next, I want to talk about differences in fungi strains. One major question in phytopathology is quantifying fungal virulence. This is an experiment using 2 fusarium strains, a commonly used lab strain, we call it moderately virulent fusarium, MVF, and one we got from a seed company that collected it from a contaminated field. We called it virulent fusarium. We did a traditional seedling assay proving the differences between the strains. In traditional methods, you take tiny seedlings and inoculate them. Then 3 weeks later, you score the symptoms. This is picture 3 weeks later. And you can see that the virulent fusarium inoculated plants were dead or wilted. The inoculated plants with MVF, with the other strain, showed also wilting, some disordered and some look healthy but do have browning in the vessels that indicate the fusarium growth inside. This test worked well, and this is a graph representing the different groups. But from my experience, it is hard to decide sometimes what group a plant is in and to find the browning in the tiny vessels. Anyway, I want to have something more field-like environment with big pots, with fertilizer, with sunlight. So I went to the Plantarray system. Using the Plantarray system, you can see that in greenhouse conditions, after 18 days, the plants still look fine. Looking at the transpiration, at day 18, we could find differences between the control group and the virulent fusarium. After 22 days, we found differences between all 3 treatments. This is showing us nicely the disease progression. And the best thing about it is that it is before visual symptoms. This is a disease score we gave visually, and it only occurred after day 22. In the last part of my talk, I will shortly introduce you to 3 other plant pathogen interactions that were monitored using the Plantarray system. The first is Phytophthora infestans that causes the late blate in potatoes. In the field, the first symptoms are usually these spots on the leaf tissue. We inoculate the plants by spraying them with a pathogen suspension and at the date where the arrow is showing. Using the Plantarray system, we followed E. E is the transpiration normalized to the plant weight. So 3 days after the inoculation, you can see here was the inoculation, 1 to 3 days after inoculation, there is a significant difference in E between the control in gray and the inoculated plants in black. In the picture that Nathaniel took, you can see the disease is not yet showing in day 3, only later. Another disease that starts in the leaves is the tomato yellow leaf curl virus. This work is of Mishra's, and he took healthy and well-watered plants, and they transpire nicely up to 800 grams. But already from day 1, virus-infected plants transpire less. Interestingly, they tested how virus-infected impacts drought tolerance. When exposed to drought, virus-infected tomatoes are more resilient than uninfected plants. the inoculated plants reached the critical point later. You see, the healthy ones got to the route earlier than the virus infected. So in the pictures, you can see the healthy plants, how they look really bad, and the virus infected are still standing strong. The last use case I will present is Wexler's work. He had an experiment with peppers that had some kind of root rot. By the end of the experiment, the shoots looked quite similar. However, the roots showed varying degrees of rot. So they classified the plants into groups of root severity. When using this grouping and looking back at the daily transpiration, they could have noticed differences between the plants that were found to be infected and the healthy plants. The difference was found already here, much before the end of the experiment. Interestingly, they compared these findings with reflectance spectrum measurements across different stages of root rot. I know many of you are in the imaging area with different drones and spectrums. I believe the first step is to fully understand how infection disrupts whole plant physiology through these continuous parameters. And by placing imaging systems over the Plantarray, we can identify physiological indices, how physiological indices impact thermal reflectance and measurements. I find this can be an important step towards better understanding field sensors and drones. To conclude, the lysimeter system enables early detection of disease, even before visible symptoms appear. Transpiration is the most sensitive physiological trait we can monitor, and this continuous system is ideal for quantitative comparisons between plants and fungi and may be utilized for breeding plant disease resistance. These days, our work is in peer review, and we would like to hear your questions and feedback. But first, I want to thank my lab members and workers and my family and friends for supporting and helping me in the greenhouse. And I'm sorry I couldn't attend the conference, but I'm grateful for the opportunity to show my work. And I want to thank you for making this possible.
Well, thank you very much for this presentation. Are there any questions from the audience? There are none. Do we have none questions, ladies and gentlemen? There is one.
Thank you very much for the talk. It was an interesting talk. I was curious if you are able to use this for the disease testing. How do you deal with the cleaning after this experiment? Because it's, this machinery. Can you explain a little bit about that?
Yes. All the pots and the sand goes to, with chlor to autoclave, and all the pots go into big big tanks with chlor, and everything is really needs a lot of work to in the end of the experiment to be clean again.